Bismillahirrahmanirrahim. This is the second lecture of the lecture series of Bioinformatics for undergrad students. In the previous topic, we talked about the history and all. Now, I explained you that there are different types of experiments in bioinformatics. What does actually that mean? That for in vitro biology we do experiments, and for the in silico biology there are also a lot of experiments. For instance, here you can see that searching, comparing, modeling, and integrating. These are the four different types of experiments we do in bioinformatics. So first of all, if we are talking about searching, that is the simplest of all, and similar to other biology or other fields, here you also perform some searching related to any question. For instance, if you want to search a nucleotide sequence, you have to go to a database where you try to identify the nucleotide sequence of your gene. So for that you have to go to the database, and then you have to perform all the process, and that can be called as an in silico experiment. Similarly, if you want to look for any protein sequence or any other information, so for that you need to do the searching, and that searching is also a type of experiment. So that's why it's the simplest of all. And but again, if you want to compare two sequences similar to what we do when we are performing different experiments, that what is the comparison between these two, the expression of these two genes, or if whether one mutation is identified in one gene and it is in one patient, and that mutation is also identified in another patient, so that for that we do any comparison. But if we are talking about the comparison at in silico level, for instance, if we want to check that how the sequence is evolved through time, here in the first upper figure you can see that here it's trying to compare four different species. For example, human, mouse, rat, cow, and chimpanzee, right? And now that is a sequence of one histone H1 protein, and after comparison you can identify that how much the sequence is conserved between the five different species. So that is also one type of comparison, and you can see that here it is mentioned that this portion is conservative. Here it is known as is saying also conservative. Here is non-conservative. You know that what does conservative and non-conservative means. We will go into the detail that the sequence which is conserved and is not changing is called conservative, and if it is changing or it is changing the in each species, then it means it is not conservative. Again, that is for evolutionary analysis. And then in the below figure you can see here that it's comparing different population and trying to identify any mutation in a specific gene. And here you can see that at one site, and this which highlighted, there is a change and between different sequence, different population. And yeah, for example, here France, Japan, USA, China, Denmark, Argentina. So there are different countries, and each sequence of TEM, probably you can say transmembrane protein, that belongs to one person, and this is a comparison of that sequence between different individuals. And here and after comparison we can identify that where actually lies the mutation. So that is also one sort of a experiment that is called comparison. Another aspect which is more very much common in protein biology, you can call it proteomics or protein structure analysis. Here we try to look for the prediction of some gene. For instance, if we have a known structure and we want to identify a structure of an unknown sequence we have, then we have to predict that. We will go into the detail, and we will explain that, and that how different softwares and tools perform these analysis. But for now you should know that when we have the known and we want to understand the unknown, we model that unknown sequence with the known sequence and try to predict based on the known information. That is called modeling, and modeling one seq structure with another and predicting the structure of an unknown sequence. So here you can see that based on the observed, and these are the predicted sequences of different proteins. For example, ELA-V4 is one protein of human, and then RASH is one protein. So these are the predicted structures and these are the observed structures. So crystal structures which are already known, right? So based on different structure prediction techniques, they perform these modeling analysis. So this is also one sort of an experiment, because in vivo, or you can say other than in silico, they use different X-ray crystallography and nuclear magnetic resonance, that is known as NMR, or mass spectrometry, and they predict the structures. But here, using bioinformatics approaches, using just the computer resources, we can predict the structure of a protein by modeling that structure with the other known proteins. So this is also one major field in the bioinformatics and major experiments we perform. Lastly, this is the most important part, not you can say most important, but most challenging. Obviously the thing which is challenging becomes important when it becomes inevitable, because this is the time of in this era there is a lot of challenges related to data, where you have to deal with a lot of data. For instance, if we just take example of one genome, for example, human genome has a genome size of more than 3.2 GB, right? So if we have 100 or 200 human genome sequences, and now we want to analyze that information. Now the first problem is that it is very labor intensive and very exhaustive, because if you want to analyze one human genome of 3.2 gigabase pair size, right? Now if we have information of 100 genomes, then that problem will intensify 100 times, right? So for that we need big resources, big computational powers, with high processors, high RAM, right? And then we have to analyze information. But if that is one challenge which we already cope with. For example, you go and you just buy a computer and you just ask them that what is the RAM of this computer, and if that is more than 8 GB, and you say, okay, I'll watch movies, I can watch, I can play games, and I can analyze the information, right? So but if you have more extensive things to do, then you ask them to improve the RAM, right? Just improve it to 12 GB, or to 18, 16, or 32, and that's so on. So the scientists, or the data analysts, or the computational scientists have identified solution to the big data. But the other part of the challenge, that is called integration. Now if we have a lot of information from different resources about similar disease, let's suppose if we say that there is one disease, cancer, right? Now we there from Pakistan we have sequenced, for instance, 100 genomes of breast cancer patients, and now there are 1,000 genomes from USA, and 150 from India, and 230 from Sri Lanka, or 800 from China. So now we have to integrate that information, right? That integration is challenging, right? So I've just told you the one of the applications, but in simple words, if we just go to the browsers like NCBI genome browser, and we want to look for the information of one gene, but that gene is information came from multiple sources. As I told you about the browsers, that browser have multiple databases. So if the information came from multiple sources, then the first challenge that will come for the integration is that how to con- come to include the information from different sources under one heading, that is one gene name, right? So the first problem would be that how to for instance, here if you can see that one problem is that what are the formats, right? Variables, what formats are used in different databases. For example, one database is using different format, other database is using different format, and then how to integrate that information. So for that there are a lot of different ways to combine those in under a uniform format, and there are different approaches, schemes that how schemas that how those different schemas are of those different data sets, and data content, what content to include and what not to include. And again, to also, if you go on the first part, that many to maintain new appearing, only 20% have a sustainable future, not easy to find them. But out of all the information, you know that whenever we talk a lot of things that are not rubbish, right? That just stupid. But there are very few things which the people who talk less are, talk better, because they try to identify the important information, just say that, right? So a lot of information is available similarly in databases, and a lot of that is not worthy enough to be incorporated into a new database. So how to differentiate between the sustainable information and non-sustainable information, that is also a big challenge, because there are so many levels of the data, and with a lot of, you know memory and a lot of storage. So how to exclude here, if you see that redundant information, that is, if one information is present more than one time, right? That is also a very uh, uh, tiresome process, that if one information came from two different resources into one browser, and that information is actually not present in both the databases, so how to exclude information from one and make it a uniform and simplified information in another browser, that is also a very big challenge for data scientists or bioinformaticians. And then inconsistencies, that is what you have to make the data uniform, that is also a very big challenge. The data is increasing with every day, and the challenge to make it consistent is also, increasing every day. So this field, the integration, right? This field, the integration, is not recommended for basic biologists. If you have good interest in data science, if you have uh, good understanding of basic statistics and mathematics, and then one programming languages, then you can come up with an idea how to integrate that information and make another tool for biologists, so that they can use the information of interest. So that is also one field. So these four are the major fields, right? So, and if we simplify that concept, that bioinformatics and this scientific method you studied since you are a child, and in that you know that first what you have to do, you have to ask a question, then you have to perform a research, then you generally come up with an hypothesis, then you test that with an experiment, and after that experiments you check whether the hypothesis is true or the hypothesis is not true, and then you report the results, right? So this whole process is also followed in bioinformatics. For instance, if you have a question that uh, I want to find that how uh, whether the expression of this gene, any gene, whether the expression of a gene is associated with cancer or not, right? This is the question. Then you perform research and try to come up with an idea that whether that is known in the literature. You go to the different hospitals and see that if any of the patients have the expression of this gene, or reported by the doctors or any in the literature, yeah, and you come up with some idea, and based on that idea you generate an hypothesis that whether A is associated with cancer or not associated with cancer, or A is associated with cancer, right? So that you do through research and come up with an hypothesis, and after that hypothesis you test that experiment. How do you will do that? Using the different data sources, databases, you download the data from of breast cancer from different databases. I will tell you how you can download those databases, those information of those genomic data from those databases. I will tell you in future. And using that data, you can check your and perform experiment and check the result that whether A is associated with the breast cancer or not, and you can come up with the hypothesis whether it is true or whether it is untrue, and then you can come up with the results. So bioinformatics also follow all those scientific methods. So all these things can be done in parallel with while you're doing your biology. So using biology, uh, you can take a lot of time, but in bioinformatics you will, that this whole process can take only, if you have a good computer, 1 to 2 days. And if you are doing it using biological approaches, that will take months. So bioinformatics can help you to come up with very good scientific hypotheses and results very quickly, and then using that information you can just validate that using experimentally, and you can come up with the conclusion. Then again, there's another term you have heard, that is called systems biology, right? So now system biology or bioinformatics or computational biology or data science and all these different fields, they obviously they have some slight differences, but uh, all of them have the same, follow the same approaches. For instance, the bioinformatics, what we do, what we perform, we try to manage the data from different resources, right? That is bioinformatics. We analyze that and come up with the conclusion. That is bioinformatics. In systems biology, there is we try to perform the systematic review, and then try to find different interactions at different levels, right? And integrate those resources. For example, DNA to RNA to protein. Now you know that's central dogma, right? DNA to RNA to protein. DNA for DNA through replication, RNA to transcription, and then protein translation for protein synthesis, right? So this whole process is called central dogma. So if you connect all those things in a systematic way, in any integrated model, that is a systems biology. Using any computational approach, if you do it, that's system biology and bioinformatics. So you cannot separate these two things. So do not get confused or get afraid of different technical terms or get overexcited that, oh, this is another field. Just be focused that here in bioinformatics we deal with the data, and for that data there are different ways to analyze that data, and there are different approaches. So one approach can be from statistical methodologies or bioinformatics approaches or systems biology. So these are different fields. Here in the at below you, I've just pasted the definition of systems biology so that you can feel that there is no major difference in bioinformatics research, computational biology, or systems biology. And yeah, this is the central dogma. So we deal with, in molecular biology, we deal with these four things. One is genomics or genome sequences, transcriptome sequence assays, and then proteomics, proteome, and metabolome. You know the difference between that. Genome actually is the structure, function, expression of all the genes in an organism, right? So, and then a transcript means one transcript, and transcriptome means the study of a whole mRNA within a cell, right? Proteome means the large scale study of proteins, including their structure and function within a cell or system or organisms. And again, for the metabolomes, right? If the whole metabolite profiles, then that is a metabolome, and if you're talking about fewer, then that will be different. So these are the four things. Another which we deal in bioinformatics at the molecular level. There is also one definition that bioinformatics is the is the computational branch of molecular biology. So what does that mean? That in bioinformatics we mainly deal with these mainly four different approaches like genomics, proteomics, transcriptomics, metabolomics, and come up with any different computational solutions, right? Again, same that mainly genomics in genes, everything comes, everything is made up of DNA. So if you talk about biochemistry, molecular genetics, central dogma, proteomics, molecular biology, any other fields all start from the genome of a cell. So we have to understand and we this first, so that we should know how to access this information, how to uh, come up with the, you can say, the how to come up with the genomes and how to analyze those genomes and come up with the answers of our interest. We will again and again, I'm repeating one thing, that question of interest or answer of interest or conclusion, because we will come up to that. We will perform different exercises during our process, and we will see that how we try to solve the questions that can be related to genomics or proteomics or any the biological field and how we solve the answer, come up with an answer, right, using computational resources. Okay, right. Next, in the next lecture, I will explain you that how the data increased with time and how we were able to manipulate that, manage that data and analyze that data, and and what are the challenges for us in future, and what are the challenges we are facing now, right? So in the next part of the lecture, we will explain couple of human genomes, so that you can have a brief idea that how much important bioinformatics is to cope with this big data of biology. Thank you. Assalamu alaikum.